Alright guys, so now we're going to continue on our DNA replication lecture. I think DNA replication is amazing. It really is. I think that it's a very, very important type of kind of topic to grasp in biology, because once you understand it, it leads you to things like PCR. You can start to understand how PCR works. You can start to understand how DNA sequencing works, which is directly related to VCV, but pretty much related to almost everything we do in biotechnology. So I think it's a very important subject for you to kind of wrap your head around. And I will warn you, it's kind of complicated, but studying it will help you to master it. And then you'll take what you learn, you'll put it in your central dogma assignment, and then you will also take it to another class and learn even more about it. This is just the tip of the iceberg, as per usual. But what I wanted to say specifically is in the Mendelian, not the Mendelian genetics lecture, the mitosis and meiosis lecture, when we were talking about the preparatory phase, interphase specifically, in the S phase, we said that DNA duplicates itself. What does that mean? And it looks so simple, but hey, guess what? It's not. There is this huge process of something called DNA replication that actually occurs, and this is how we actually replicate a chromosome into two sister chromatid that are attached at a centromere that are ready to get pulled apart in mitosis, or ready to form tetrads with their homologous pairs in meiosis 1, and then those get pulled apart, and then the sister chromatid get pulled apart. So I just wanted to warn you and tell you that when we said chromosomes get duplicated, what does that mean? This is what it means. Those chromosomes go through the process of DNA replication. Okay, but we know that we have 46 molecules of DNA in your cells. 46 molecules being 46 chromosomes. But what I think is really awesome to put things in perspective is that of those 46 chromosomes, all of the nucleotide bases in the specific sequence that make up you and make up me, if we were to take all of those codes and put them into page by page, type them out, A, C, T, 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 A, B, or A, G, something like that. If we were to do that continuously for all of our chromosomes, it would fill 1,400 biology textbooks. So that's the textbooks that you have right in front of you. Those are not for the weak-hearted, really. They're very intense textbooks. I don't know, some of you might even have the half edition of the textbook. So that just kind of puts things in perspective for you, that we are made up of millions and millions and millions of base pairs that code for all of the proteins and all of the RNAs in our body. But what is even more miraculous is that it only takes cells a few hours to copy all of the genetic material, and there's relatively very little error in comparison to how much it does, how much it replicates, which is a huge feat. There is something called an error rate of 1 to 10 billion. So there is one error made in every 10 billion base pairs read. So that's awesome. Because if not, we would just be like bacteria that are completely mutating all the time and changing and doing that. I decided that we're not going to get into the repair mechanisms and why we have this 1 in 10 billion error rate, because you just have a lot on your plate, and I know that those are very important in other classes, and you will get to them in other classes. So I think that just what I'm very concerned about you understanding is just the overall process and mastering that before you move on to something else, because I think that if you don't master this, and I kind of overwhelm you with the repair mechanisms and a lot of the other stuff that your chapters talk about, you're missing out on the kind of the fat of it all, the juicy part of it all. So I want you to kind of understand this stuff. Okay, so today what we're going to look at is we're going to look at the process of DNA replication, but what I want you to keep in mind is, one, all of the enzymes and different names of things, the whole language associated with DNA replication, and then I want you to also keep in mind leading and lagging strand replication, how they're different, because you will see how they're different. But keep this in mind when you are thinking about your central dogma assignment. Okay, let's move forward. So each DNA has a start site. This start site is called the origin of replication. Each chromosome has an origin of replication. So each DNA chromosome has an origin of replication. So we're showing you in these pictures the difference between E. coli genetic material, which is in a circular ring, and chromosomal genetic material, which is linearized. Yes, it's coiled up into different things, but it's linearized before it gets duplicated and then coiled up into metaphase chromosomes. So let's look at this. We have specific replication machinery that recognizes these specific start sites, these origin of replications. DNA replication occurs in both directions until the entire chromosome or the entire plasmid or the entire circular bacterial genome is replicated. This concept of going in both directions, these little arrows that you can't really see that are pointing in both directions, is called bidirectionality. So this is the ability for us to replicate in two directions away from the origin of replication. So one key difference between prokaryotes and eukaryotes is that prokaryotes typically only have about one origin of replication in their genome, whereas we have about 1,000 origins of replications in our chromosomes. We have just tons and tons and tons of them. And what this picture is showing you is that one, E. coli DNA is very small in comparison to all of the chromosomes in every cell of our body. But what this is showing you is that because we have multiple origin of replications, these little replication bubbles start to open up, and then they start pushing towards each other, and soon they push into each other and form, those circles start to kind of expand when they meet to each other. So what is the significance of this? So if you have multiple start sites, these bubbles can fuse together and create, they can start replicating this strand in a way faster rate than just having one origin of replication, because these bubbles will eventually merge together, all of them. Okay, so this is the start site of DNA. So prokaryotes have one origin of replication, eukaryotes have many. Okay, so we're going to go over the overall process of DNA replication, just kind of the players that are involved and the language that's involved, and then we're going to go into leading and lagging strand replication. We're going to watch a movie right before we get into leading and lagging strand replication. It's actually a really good movie, and I hope you watch it a lot. But it helps to kind of put everything in your mind, is how it works. Animations really help for you to see what's happening, because pictures can only tell you so much. So let's get into it. So in DNA replication, we talked about there being origins of replication. And in those origin of replication, specific replication machineries opens those origins of replication up. The little bubbles that they create, these little bubbles that replication machinery creates, is called a replication bubble. But on either side of this replication bubble, this side is not shown, But on either side of this replication bubble, there's something called a replication fork. Now, this is a very important kind of landmark of DNA replication. So keep in mind the origin of replication, which is right here, and the two replication forks that are right there. So one of the major enzymes that is involved in the DNA replication process, the unwinding process, is called helicase. Helicase is like a wedge, and what it does is it wedges in between these double-stranded DNA of the parental strand, and it breaks those hydrogen bonds and pushes through this DNA, opening it up. So we saw this happen in with RNA polymerase. RNA polymerase didn't need this molecule called helicase to do this for it, because RNA polymerase, because of its structure, is already like a wedge. So when we are doing transcription, it opens up the double-stranded DNA molecule at a specific gene, and it reforms at the other end of RNA polymerase as it's pushing this way. Whereas DNA polymerase doesn't do that. DNA polymerase requires this enzyme called helicase, part of the replication machinery, to push open this double-stranded DNA. What else I wanted to say is I earlier said the parental strands. So don't forget that we were talking about that DNA replication, the model of DNA replication is the semi-conservative model, saying that both of these parental strands split open, and these single-stranded DNA molecules serve as templates for the new daughter strand that gets created. So there would be one for this guy and then one for this parental molecule of DNA. So helicase is the wedge. One thing that's interesting about DNA replication that we don't see in the central dogma in transcription is that these proteins, called single-stranded binding proteins, bond to these single-stranded DNA. What they do is they prevent these two strands, these two parental strands, from re-annealing or re-coming back together. So single-stranded binding proteins bind to single-stranded template strands and prevent the rebinding of the parental strands. So that's what they do. They help to keep these two away from each other. They're not to keep them away from each other, but to keep them from reforming, recreating those hydrogen bonds between their nucleotides. Okay, what is another enzyme that's important to this whole unwinding process? So this is kind of really difficult to see, and the way that your book explains it is it leaves a lot to be desired. So I posted a video. Realize that in DNA replication there's two types of topoisomerases. There's topoisomerase 1 and topoisomerase 2. We're not teaching you that. All I really want you to know about topoisomerase is this. But I want you to know that as helicase is unwinding this double-stranded DNA, these two parental strands, and these single-stranded binding proteins are preventing these strands from coming back together, which is different than RNA polymerase. RNA polymerase is right here, wedging in between, and then these directly kind of reform after RNA polymerase is moving through this. This is different than DNA polymerase because these single-stranded binding proteins prevent the re-annealing of these parental strands. So what I need you to know about topoisomerase is that it untwists the double helix, and so untwisting the double helix creates a strain. So when you watch that video, you'll see that as we untwist DNA and keep it untwisted, there is strain building up in this double helix molecule right here. This strain is relieved by an enzyme that introduces cuts. This enzyme is topoisomerase. Whether it's one or two, I don't really. It doesn't matter for me for you to understand that right now. I just want you to know that topoisomerase, as a class of enzymes, what they do is they introduce little cuts in this upstream of the replication fork, which relieve pressure. so that it introduces a little cut and then it relieves that, and then it reseals that cut. This whole process kind of relieves strain so that this can move forward. That's basically the level that I want you to understand it at. So what you see here, though, is I talk about DNA gyrase and topoisomerase. These two enzymes are analogous to each other. They do the same thing. But DNA gyrase is an enzyme that is only found in prokaryotes. Prokaryotes do not have topoisomerase, they have DNA gyrase. Humans have topoisomerase. Why is this important, and why did I make a point of putting both of these in here? So prokaryotes, eukaryotes. Why is it important? Because in microbiology and in your nursing classes, I don't know which ones, but you will learn about specific antibiotics that target bacterial DNA replication. So it blocks bacterial DNA replication without blocking human DNA replication. So this area, this difference between humans and bacteria, is something that we can target bacteria. We can target bacteria cells without targeting human cells. So this lays the groundwork for a very big class of antibiotics that you'll learn about. But specifically, this idea of targeting bacteria cells and killing them off without targeting human cells, because you got to remember that bacteria, especially pathogenic bacteria, are in our body. So if we take medicine, that medicine is going to affect us and it's going to affect the bacteria. So we need to find antibiotics that are that selectively target the bacteria without targeting us. So if we can find drugs that target DNA gyrase without targeting topoisomerase, we can shut down bacterial DNA replication without shutting down the DNA replication in our own cells. So that's very important. This idea of doing this targeting of bacteria without targeting human is called selective toxicity. Okay, so kind of something that we've been neglecting to talk about is that 3 to 5 prime orientation. So we know that transcription occurs in a 3 to 5 prime direction, and we also know that DNA replication only occurs in a 3 to 5 prime direction. But what do we see here? There is 2 strands of DNA. DNA is double-stranded. This strand is 5 to 3, so what is produced going this way is a 3 to 5 prime orientation. But how about going this way? So if I were to build a strand of DNA from a newly synthesized strand from this template strand, and DNA polymerase was moving this way, what would happen? Well, it would be in a 5 to 3 prime direction. So that's not what happens. That cannot occur. We just know that, and I told you to just know that as a general rule, that DNA replication and transcription only occur in a 5 to 3 prime manner. So let's read this little problem and this little dilemma and then look at the solution. So a specific enzyme called DNA polymerase, so it's just like RNA polymerase, it creates polymers from monomers. So the monomers being DNA nucleotides, it attaches them to form a polynucleotide. It creates a dehydration reaction that creates those phosphodiester bonds that link nucleotides into a polymer or a polynucleotide. This is what DNA polymerase does. Polymerase, polymer, and then ACE refers to an enzyme that does this polymerization action. So a DNA enzyme called DNA polymerase in the elongation step, which is the next step after unwinding, cannot initiate the synthesis. It cannot initiate synthesis and needs to add nucleotides to the 3' end of an already existing chain base paired to the template strand. That sounds like a mouthful. But what we're going to see in the next slide is there's an enzyme called DNA polymerase. But what is different between DNA polymerase and RNA polymerase is that DNA polymerase needs to add nucleotides to an already existing strand. It needs to add it to the 3' end of this already existing strand. So it's going to lay down complementary bases one by one this way, but it has to have a 3' end to build off of. So that's what this means. Realize that when I'm saying it has to have a 3' end to build off of, it needs to build off this 3' end, but it's still replicating DNA in this 5 to 3' orientation. Sorry that I messed that up. So what is the solution for this to occur? We need primers, and we've talked about primers in the PCR reaction. So what I kind of want you to understand about science is it's a process. You need to learn the basics before you can start to create different higher level forms of science. So that guy that we talked about from San Francisco that created the PCR reaction while he was driving in his car to his cabin in, I think it's Mendocino County in the hills of San Francisco. He needed to understand this whole process of DNA replication before he could even consider the PCR reaction, which has been monumental in all of biology. So understanding the basics, I know this stuff is kind of tedious and hard to master, but once you master it, then you can start to apply it to different things and understand different things. I'm going to tell you that once you understand DNA replication, we're not going to tell you this, but once you understand or teach you this, we're not going to teach you this, but once you understand DNA replication, once you understand the PCR reaction, then you can start to understand something called DNA sequencing, and you can start to see how scientists have developed a way to sequence DNA. But that's down the road for you. That's down. We're not going to teach you that this semester, but you will learn it in specific classes. If you ever take a genetics class, you will learn how this process of DNA sequencing is related to replication and related to PCR. Okay, that's all I'm going to say about that. So the solution of this problem that is posed up here is primers. Primers are needed in DNA replication. Were primers needed in transcription? No, they were not. We did not need primers because RNA polymerase was an awesome molecule that could do all of this stuff on its own. It didn't need to add to a 3' end of something. It could self-start itself. So that's a very important difference between DNA replication. So let's go to the next slide. So how do we solve that problem? Primers. But there's a specific enzyme called primase that does this for us. We can start to see just off the name, what does primase do? Primase is an enzyme that creates primers. So what does it do? Primase starts elongating a complementary RNA, not DNA, RNA chain from a single RNA molecule that's just kind of swimming around in the nucleus, adding more and more RNA complements using the parental DNA as a template strand, producing primers, usually 5 to 10 base pairs long. So basically what that says is in the nucleus there's just these RNA nucleotides. They are called ribonucleotide triphosphates. So in DNA polymerase, or in DNA replication, we're usually concerned with deoxynucleotide triphosphates. So deoxy-A's, deoxy-T's, deoxy-C's, and deoxy-G's. Primers, and what we use in primers for this application is ribonucleotide triphosphates, RNTPs, are ribonucleotide A's, T's, U's, and A's, U's, C's, and G's. Sorry, I'm confusing myself here. But basically what this is saying is that there's these RNTPs just floating around in the nucleus. These nucleotides just get laid down by primase. So primase lays down nucleotides upstream of the origin of replication, And this is the red color that you see. It's an RNA primer, so it's just laying down this nucleotide, matching it to its complementary base on the parental strand, so that it makes this little kind of 5 to 10 base pair long primer. When we create this primer, then DNA polymerase can come in and start to lay down DNA nucleotides on the 3' end of this primer. So this is solving the problem that DNA polymerase creates, primase and primers. Okay, so now we're going to actually start to talk about DNA polymerase and the elongation step, or the polymerization step, creating a polymer. And just keep in mind all of the basics. The basics are still occurring here. Nucleotides are coming together, so monomers are coming together to form polymers, the polymer being a polynucleotide. So what reaction occurs? DNA polymerase is facilitating a dehydration reaction that creates a phosphodiester bond in the sugar phosphate backbone of the nucleotides and of this polynucleotide. So that is what is occurring. DNA polymerase is an enzyme that catalyzes the synthesis of new DNA by adding nucleotides, DNA nucleotides, deoxynucleotides, to the 3' end of a pre-existing chain, aka the primer, laying down nucleotides that are complementary to the template strand, or the parental strand of DNA. So this is the template strand. So DNA polymerase is laying down these nucleotide monomers that are complementary to the parental strand. Okay. So what is important about the difference between prokaryotes and eukaryotes? Well, one is prokaryotes only have a few DNA polymerases in them. So bacteria only have a few DNA polymerases in them. Whereas eukaryotes, we've found 11 different types of DNA polymerases. So when I took some of my courses, I needed to memorize all 11 of them and be able to explain what they did. That was a mess and a half. I did not do well on that exam. I didn't do bad, but that's besides the point. But what I want you to be aware of in our class, what is important is that you understand two types of DNA polymerases that are found in eukaryotes: DNA polymerase 3 and DNA polymerase 1. Denoted these. These are important, and we'll start to see the kind of ornate differences between these two types of polymerases. Okay. Here's another important difference between prokaryotes and eukaryotes, or prokaryotes and humans, bacteria and humans. The elongation rate, so the rate at which these nucleotides are put down by DNA polymerase, is drastically different. So in bacteria, they lay about 500 base pairs down per second. That's really freaking fast. Whereas in humans, we lay about 5 base pairs per second, which is still pretty fast. But what is the significance of this kind of disparity? The significance is that bacteria are laying these down very, very, very fast. So they make a lot more mistakes than human polymerases make. Why is that important? Well, this leads to bacterial mutations. Bacteria have a ton of mutations that occur at a very quick rate, whereas mutations are very slow to come about in human genetics. This is important because we always talk about bacterial, well, we don't talk about it, we haven't talked about it yet, but bacterial mutations. So bacterial mutations, so think about it, if you give an antibiotic to a person and these bacteria are replicating like crazy, what happens is, what is logical to happen is that one bacteria might have one or a few mutations that occur when it's replicating its genome. Some of those mutations might kind of be beneficial for the bacteria. It might help the bacteria to fight off the antibiotic. It might help certain things to occur that help the bacteria to thrive. So what would happen in that environment is all of the rest of the bacteria will start to die, but the specific bacteria that has the mutation that is beneficial will live. That bacteria will replicate in a crazy fast exponential rate, and then pretty soon in your body there is this large amount of antibiotic resistant bacteria. That's the significance of this. Okay. So here I put this in bold because this is a little bit different than you are used to seeing. Nucleotides in DNA replication and in RNA replication, the reason we've been calling them deoxynucleotide triphosphates and ribonucleotide triphosphates, which is different than what we initially taught you, is that nucleotides are like ATP. They're very similar to ATP in the sense that they have a sugar, a phosphate, and a nitrogenous base. But ATP has 3 phosphates, and the same occurs in nucleotides, which you didn't know that before. Before, we just showed you the sugar, the phosphate, and the nitrogenous base. Now we're showing you the sugar, the phosphate, the nitrogenous base, but there's 3 phosphates attached to this nucleotide. This is how they naturally occur, and this is very important. Why is it important? So the DNTPs, DATP, DTTP, DCTP, DGTP, these are DNTPs. The D denoting deoxy, and then the specific C, G, T, and A denoting the nitrogenous bases. But why is this important? So just like ATP, these phosphates all have negative charges that are bound together. This creates a ton of potential energy stored in these bonds. Those phosphates are constantly experiencing repulsion, but those bonds are holding those electrons together in those bonds. So why does ATP have so much energy? That's why. Why do these nucleotides have energy? This is why. But what's very important is that why do we need energy in nucleotides? Well, we need energy to occur to move forward the elongation step of this process of DNA replication. What is probably not clear in your book is that, and what I want to retouch on, is that taking a single nucleotide and building a polymer of nucleotides is an anabolic reaction. We know that anabolic reactions are endergonic, and they need energy to move forward. So this is the energy that is required for this anabolic reaction. So what's happening is once DNA polymerase lays this nucleotide down, it facilitates this dehydration reaction that connects this sugar and this phosphate. But what happens is this pyrophosphate just means 2 phosphates linked together, this pyrophosphate pops off. What did this pyrophosphate do? Well, this pyrophosphate has a way of breaking down into 2 inorganic phosphates, but when you break this bond, energy is released. This energy is utilized to move the elongation process forward. So this is very important. Okay, so now we kind of glossed over this, but now we got to go back and revisit it, because you're kind of seeing how DNA replication works, but you're not really seeing the whole picture. Why? Because DNA is in an anti-parallel conformation, meaning that if you look at this strand is in a 5 to 3 prime conformation, whereas this strand is in a 3 to 5 prime conformation. What does that mean? They're anti-parallel. They run opposite of each other. So that creates a very huge problem, because DNA polymerase can only work in a 5 to 3 prime manner. So yes, this strand works in a 5 to 3 prime manner, but what about this guy down here? What happens on this guy? So what these are termed is, this one that's in the correct orientation for DNA polymerase to work is called the leading strand, and this one on the bottom that's kind of wonky is called the lagging strand. And we're going to start to see how leading strand and lagging strand have two different replication models that occur, and it's important for you to understand both of them. So before we move forward, I want to kind of just watch this video. I will talk through it and kind of help you to see things, but then we'll start to go through the slides of leading strand and lagging strand replication. But it's very important for you to watch it first because it's not very intuitive. It's not very easy to see, and it's kind of difficult. So watch this first, and then we'll go through it. Oh, this is the video of topoisomerase. Please watch this. It's already on D2L. Okay. And I'll post this one on D2L as well.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates
So that's
for making
the replication
new strands.
bubble.
The result is a replication bubble.
These are the replication
The bubble
forks.
grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue.
Remember the semi-conservative
Because
model.
strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces.
So we're going to look at the differences between the leading and the lagging strand, and why they're even called this.
First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands.
Serves as a template. The parental strand serves as a template.
Each piece of the lagging strand begins with a short segment of RNA, shown in red.
Pay attention here, guys.
A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA.
Let's see how it's...
When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
So it's removing the primer and putting down DNA nucleotides. So it removes the RNA nucleotides and puts down DNA nucleotides.
However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together.
We learned about this in lab. The glue, it glues the backbone together. DNA ligase.
Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules.
That become
Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Okay. Now let's go through it. I hope that was helpful. Okay, so let's go through leading strand replication first because it's a lot easier. So just keep in mind that leading strand replication occurs in a 5 to 3' direction. The reason it's termed leading strand replication, we'll go over that in a second. But remember that there is this replication bubble. Inside of the replication bubble is the origin of replication, this little guy right here. That's the center. Right here and right here, these are replication bubbles where helicase is pushing outwards and pushing outwards, breaking those hydrogen bonds. So what is occurring? So these are the two parental strands that serve as templates for the newly synthesized DNA. This is the semi-conservative model of DNA replication. Okay, so in leading strand replication, DNA replication is already in the 3 to 5 prime direction. So leading strand replication, what I want you to see, is moving in the direction of the replication fork. Because if you look at it in this bigger picture, this DNA, this parental strand, is in this 5 to 3 prime direction. But if you're moving this way towards the replication fork, what is being replicated is in a 5 to 3 prime direction. So this is in 5 to 3 in this direction, so going right. But this one is in a 5 to 3 prime direction, moving towards the replication fork. That's really important. In leading strand replication, DNA replication is already in a 5 to 3 prime direction. This is in and 1 and 2. That's what I'm showing you right here. DNA polymerase remains in the replication fork and can continuously add nucleotides to form a new, that should not say template strand, a new strand or a daughter strand. So DNA polymerase right here, DNA polymerase 3, is adding, so what happens is helicase is breaking through over here. What happens is right upstream of the origin of replication, primase puts down an RNA primer. Then DNA polymerase 3 adds nucleotides to the 3' end, but since it's in a 3 to 5' orientation, it just continuously moves with the replication fork, moving with helicase down this way, adding complementary bases to the template strand. So this is how DNA leading strand replication occurs. It occurs continuously, and only one primer is needed. Only one primer at the origin of replication is needed, because what happens is it moves all the way down to the end of the DNA molecule. So it just is this continuous process that occurs. So what your book doesn't tell you is this primer needs to get removed. You can't have DNA with patches of RNA in it. So that needs to be removed. So how do we do that? The primer's RNA nucleotides are pulled out by a specific molecule or a specific enzyme called DNA polymerase 1. So the primer's RNA nucleotides are pulled out one by DNA polymerase 1 and then replaced with DNA nucleotides. So that was that other molecule that's pulling out these RNA nucleotides and then replacing them with DNA nucleotides. Then what happens is, and this is not shown in this picture, but another enzyme called DNA ligase. So once this primer is replaced all the way to this guy with DNA nucleotides, there's still this kind of gap between these two nucleotides that needs to be sealed. So what happens is that glue, that molecular glue, DNA ligase comes in and seals that gap. So that's it. What did I write? DNA, what did I write? Then comes the sealing, the backbone newly formed. DNA. So it joins the sugar phosphate backbone of the DNA, the former primer, and the new leading strand. So that's what happens in this leading strand replication. Now let's start to look at the lagging strand replication. So back to this big picture up here. Leading strand replication goes with the replication fork. So on both sides of the origin of replication, it's going with the replication fork. It works in the direction of the replication fork. Lagging strand replication is different because it still has to copy things in a 3 to 5 prime direction, but what happens is lagging strand replication moves in a 5 to 3 prime direction as well, but it moves away from this replication fork. So both of these guys are moving away from the replication fork and towards the origin of replication. So that's what's happening in lagging strand replication. Okay, this is my next slide, and I don't know why I got ahead of myself. Okay, lagging strand replication. Remember that DNA replication is in the 5' to 3' direction only. DNA polymerase 3 must work away from the replication fork and towards the origin of replication. So away from the replication fork and towards the origin of replication. Away from the replication fork and towards the origin of replication. But do you see how it's in this kind of segmenty, choppy type of way? There's a reason for that. So the reason we call it lagging strand replication is because these two processes, both leading strand replication and lagging strand replication, are occurring simultaneously. But the lagging strand is much slower. Why? Because we have to add more than one primer to accomplish this goal, to accomplish this type of replication. We have to add these little red bits, are primers. We have to add multiple primers, hundreds and hundreds of primers to make this process move forward. Whereas in leading strand replication, we only need one primer and it works continuously. So essentially, leading strand replication works faster, and lagging strand replication is lagging behind leading strand replication. So that's why it's called that. Okay, now let's look at the whole process. So realize these two slides are the same picture, and they're different pictures. So this is step 1, 2, and 3. This is step 4, 5, and 6, and those correlate. But these words are the exact same thing. I just did this for show. Okay, so lagging strand replication is fragmented. Why is that important? Okay, let's look at the whole process as a whole. So upstream of the origin of replication, lagging strand or primase adds RNA nucleotides to create little primers. It also jumps up here a little bit upstream of the origin of replication and adds another primer. So essentially what this did is you were seeing that this isn't if it was going to go towards the replication fork this way, it would be replicating in a 3 to 5' direction, which would be wrong. DNA polymerase can't do that. So it has to replicate in this 5 to 3' direction. So this is why we lay down multiple primers to accomplish this replication that is away from the replication fork and towards the origin of replication. So primase adds the primers. DNA polymerase adds nucleotides, DNA nucleotides to the 3' end of the primer. So here is DNA polymerase adding nucleotides to this 3' end of this primer. So it essentially adds these two primers, then fills in the gap. And what is this little segment, this fragment called? This is something called an Okazaki fragment. An Okazaki fragment is just this fragment of DNA that occurs in these patchworks. The reason it's called Okazaki is because this is the Japanese scientist that discovered this lagging strand replication and gave the name to these fragments. So this is why we call them Okazaki fragments. Not very easy to memorize, but cool. I wish one day that my name gets turned into something cool like this that's in tons of textbooks, but we all know that that's not going to happen. Anyways, so this is now called Okazaki fragment 1. So from the RNA primer for fragment 1 and this primer down here, it fills in this little space with DNA nucleotides. Okay. So after reaching the next primer, DNA polymerase detaches. Then what I'm saying here is Okazaki fragment 2, which is going to be up here, is primed, and that's in the next step. So let's move down to this one. So it's primed up here. So a primase comes over here and puts in a primer here of RNA nucleotides, making this kind of hole between the two primers. So then what happens is DNA polymerase adds DNA nucleotides to the 3' end of this primer and fills in this gap. and this becomes Okazaki fragment 2. So after reaching the next primer, DNA polymerase detaches, Okazaki fragments is primed. Okay, DNA polymerase. So this is important. We're on step 5 now. Sorry, I had to look at where we were. But in step 5, what happens? So this is a different DNA polymerase. This is DNA polymerase 1. What does DNA polymerase 1 do? It replaces the primer. So DNA polymerase 1 replaces the RNA primer of DNA. How? Now that Okazaki fragment 2, so this is Okazaki fragment 2, is laid down, now that this is here, there is a 3' end here to this primer. So DNA polymerase 1 can lay down, excise these, or when I say excise, I mean take out these RNA nucleotides and put in DNA nucleotides, adding them to the 3' end of this Okazaki fragment 2. So that's what happens. Then once these are replaced with DNA molecules, then an enzyme called DNA ligase seals up the backbone. And in this discontinuous process, we can make lagging strand replication. So that's how this works. And I think it's really cool. How is it different? Lagging strand replication is fragmented. Leading strand replication is continuous. Okay, now we need to move on. Okay, so let's revisit the organization of genetic material. So this is the same thing that we kind of been going over all year. DNA has a negative backbone. Why? Because DNA is made up, the backbone of DNA is made up of sugar and phosphate groups. Phosphate groups carry a negative charge. This gives DNA a negative charge as a whole. Why is that important? Well, there's these proteins in our body called histones that have a positive charge. So the negative charge of DNA is attracted to the positive charge of these histones. So it wraps around these histone proteins. So there's a better picture here, and I kind of want to show it to you. This is in your book. So DNA is negatively charged. It's just showing you the backbones right here. These histones, which you didn't know about, but these histones come together and form something called a histone octamer. What does that mean? It just means that there's 8 histones that form this kind of little bead that come together. That's all you need to know. But I'm not going to test you on that. I just want you to know that the negative DNA wraps around these positive histones. And what is this structure now called? This is the bead and thread structure that we were talking about. These are the nucleosomes. The nucleosomes are a more highly organized structure of genetic material. Then what happens is these nucleosomes start to wrap around each other and form a more condensed type of structure. This is now starting to be called chromatin. Chromatin is a thicker, more higher organized structure. Then what happens is, if you see from this one, this is a 10 nanometer fiber, and then we move to a 30 nanometer fiber. So this is a 30 nanometer fiber of chromatin. This is how it usually exists in your nucleus. So in the nucleus, when I say that there's a jumble of chromatin in it, it usually is existing in these kind of 30 nanometer fibers that are still kind of easy to get to by the replication machinery and the transcriptional machinery. Then what happens is this strand, this rope of chromatin, even condenses further. It loops around itself even more and more and more, and you can see this kind of theme where it's looping and making these more organized structures. And then what happens is it turns into this structure that we know as the metaphase chromosome. So realize, let's connect us back to mitosis and meiosis. We say that DNA is replicated. Yes, DNA is replicated, but then what happens in the prophase 1, in prophase of mitosis and prophase 1 of meiosis, what happens is that more compacting organization starts to occur. that genetic material gets more and more and more compact, so that by metaphase, either metaphase or metaphase 1 in meiosis 1, chromosomes start to look like this. So what we see happen in DNA replication is, here is DNA, here is a nucleosome. We see these little strands that replicate each other. That's what happens in DNA replication. There is a replication of DNA. So now you have two identical strands. These two identical strands are called sister chromatin, but in the process of prophase 1, these sister chromatins start to condense and start to condense and start to condense and start to condense in this way, and then by metaphase, this is the structure that we see. The reason that we taught you the other way first, we just always were concerned at looking at things like this metaphase chromosome. The reason we taught you this is because there was no way for you to see how the process of mitosis and meiosis occur without looking at structures like this. If you just saw a jumble of crap in a nucleus, you would never know what is occurring, and it would be very hard to visualize for you. Okay, now, now let's move on to this. This in red is something that I'm adding different. So I told you that chromatin is a higher order complex of organized nucleosomes, usually existing in the nucleus as a jumble of strands. So this is what we first initially thought it was just this jumble of strands, kind of like a knot of strands inside of the nucleus. That turned out to be incorrect. What we started to realize is that the chromatin kind of have some organization to them, and a little bit more than we initially anticipated inside of the nucleus. So this is actually not a jumble of chromatin. What is in the nucleus is the chromatin is looped, is attached to the nuclear lamina, which is just kind of the skeleton of the nucleus that is keeping it in this circle structure. So the nuclear lamina is in this kind of, it is close to the nuclear membrane, but it helps to organize the these jumbles. So they're kind of in, this chromatin is kind of in an organized fashion. And specifically the nuclear matrix also, which are proteins inside of the nucleus, kind of analogous to the cytoskeleton of the cell. These also help to organize the chromatin. Why is this important? Because if they're organized and they're not in like this knot-like structure, then we can easily replicate them or transcribe them. That's why this is important. But I didn't teach you this at first because, you know, it wasn't really important to what we were learning then. So now it's important now. So there's that. Okay. Now you have all of DNA replication, all of the lecture for transcription and translation, and all of that, and protein modification. You have almost everything of this course. You have learned almost everything. Now let's start to see how DNA directly affects proteins. So when we say mutations, what does that mean? Mutations are often at the DNA level. So what happens when we get a mutation, or when bacteria get a mutation, or when fungi get a mutation? What happens is there's these types of mutations that occur. One type that I want you to be aware of is a point mutation. Point mutations are when a single nucleotide, DNA nucleotide base is kind of laid down wrong. So in the replication process, when the newly synthesized strand, the daughter strand, is being made, sometimes DNA polymerase makes a mistake and lays down the wrong nucleotide. That can happen, and it does happen. So when one nucleotide is laid down wrong, this is a point mutation. So in point mutation, there is these different categories of point mutation. It's called the silent mutation, the non-cis mutation, and the frame shift mutation. Okay, so what is the silent mutation? This picture is very small and kind of hard to see, so let's move to this guy. but this is the original correct strand of DNA. This is what it's supposed to be. T, A, T, T, G, G, C, T, A. You can read it. This is the original sequence of DNA. So what is a silent mutation? So this is having you connect all of the ideas that we've learned about DNA, mRNA, proteins. So what is a silent mutation? So a silent mutation is a change in the nucleotide sequence that does not change the amino acid specified by the codon. What does that mean? So there's a mutation. It's supposed to be a T, but somehow there's a C put down in its place when it's being replicated. Then it goes to transcription. Transcription shows you that what am I trying to show you? Oh, so transcription. In transcription, this went from a UAU to a UAC. So if we pull back out our codon table, let's do that. If we pull back out that codon table that I gave you, I think this is the one you have on your notes. So let's go back to this guy. So UAU and UAC. So you read first base U, A, U. Then U, A, C. These both code for the amino acid tyrosine. So this mutation is kind of negligible. It has no effect. It's silent because either one of these codons code for tyrosine. What is this idea though? This idea that multiple codons can code for one amino acid is the idea that the genetic code is redundant. So we said that there were 64 total codons. There were 64 total codons on this chart that code for different things. For instance, all of these codons code for threonine. There's 4 different codons that code for threonine. There's 4 different codons that code for proline. This is the idea that the genetic code is redundant. So what happens in a silent mutation is there's a mutation that occurs, but it doesn't have any really effect because the primary structure of this polypeptide is the same. It did not change. It's still threonine, and it's still threonine here. Okay, what is a nonsense mutation? So a nonsense mutation, what is this showing you? Is that when, let's read the thing. A change in the nucleotide sequence that results in an early stop codon. So that's what a nonsense mutation is. What's a stop codon? It terminates translation. So what happens is it's showing you that this was the right genetic material. This was an error. So what happens is this gets transcribed into mRNA, and that mRNA leaves and goes to a ribosome, and then it starts to lay down the amino acids in a polypeptide chain. So what happens is this, instead of being coded for what it's supposed to be coded for, a UGG that codes for a tryptophan, instead of that, it becomes a UGA. One single point mutation, one change in the base, leads to a stop codon being placed down. This stop codon codes for a releasing factor that breaks off the polypeptide. So essentially what this does is if the protein's supposed to be, a lot of polypeptides, a lot of amino acids in a chain, we say it shortened this protein, or it truncated it, made it a lot smaller than what it was supposed to be because a stop codon was put down instead. These have very detrimental effects. So if you get a mutation like this is probably going to lead to some sort of disease or disorder. This happens a lot. There's multiple things you can look up that say nonsense mutation disease. If you put that into Google, you would find a ton of things. Okay. The last one. This is also a very detrimental mutation. Why is it called a point shift mutation? So a category of the point shift mutation is called a frame shift mutation. This is the addition or deletion of a nucleotide. So right here we're showing you an addition of a nucleotide. So these are called additions, insertions, or deletions is what we usually call these. But these lead to frame shift mutations. What is that? So because we added an extra nucleotide, this throws off that reading frame that we were talking about. It adds an extra nucleotide in the mix. So what happens there is that this reading frame is thrown off, and it changes the codons. It changes the 3 triplet base pairs. So it moves it down a little bit so that each one of these codons are kind of thrown off one place value. So now it starts to lay down different amino acids. So it gives you a completely different protein, which is kind of just not really functional, and it's not really, it won't help you in any way, really. So these are very detrimental as well. Okay, so that's it. That's all I wanted to go over. This was a long lecture. I'm sorry. Okay, I hope you guys can use this to study, relax, realize that this stuff is not going to be until your final. So you have quite some time to come into office hours. You have quite some time to read the chapter, read the whole chapter, but specifically focus on I'm only going to test you on the things on my slides, but I think all of this stuff is important, and we just don't have the time to get through all of it. But understanding this and mastering this will really help you in later classes. It'll really help you in throughout your biology-based career. So I hope that this helps. I wish you luck on your final and on exam 4. You'll get your central dogma. You just have those 2 last cells. I hope also that this stuff starts to put VZV in a little bit more of a context for you. Now that you're starting to understand how DNA polymerase works and how primers work, we can start to see how primers targeted a gene, or they didn't actually target a gene, they targeted a promoter region of VZV. So those 2 primers, the forward and reverse primers, are seeking out those promoter regions. When we add those primers, what we effectively do is we add a 3' end that DNA polymerase can start laying down nucleotides on. That's how we are able to amplify the DNA. And we go through these cycles of the denaturation, annealing, and elongation, creating more and more and more and more of this gene product, this PCR product. So it's not a gene product, sorry, it's a PCR product. But that's really important. But to understand that science, you need to understand DNA replication. And I hope that this lecture helps you to understand that. Okay, well, good luck. Goodbye.